next.
A very good afternoon to one and all. I, Dr. Rajan Kalankar, Assistant Professor Westcott, welcome you once again to the afternoon session of this AICTE-sponsored ATAL Faculty Development Program on Research and Innovation Ecosystem in Academic Need of the Hour, organized by Vivekanand Education Society's College of Pharmacy, Mumbai. It will be my pleasure to invite and welcome respected Mrs. Maharukh Rustomjee Madam. I request respected Dr. Supriya Shidai Madam, Principal Westcott, to welcome Mrs. Maharukh Madam by giving the sampling as total of blood.
I'm privileged and honored to welcome you.
Thank you so much, Madam. I would like to take this opportunity to introduce you to the today's speaker for this session. Mrs. Maharukh Madam is pharmaceutical design scientist, idea innovator, value builder with more than 35 years of experience in the industry as a technocrat and entrepreneur with a core competency in formulation research and drug delivery system innovation, phytochemical based healthcare product, intellectual property, manufacturing, regulatory sciences and drug delivery business covering all geographic territories. She holds master degree in pharmacy from ICT Mumbai and a certificate in farm technology from University of Ghent, Belgium. Respected Madam is currently the founder and managing partner of Amaterasu Lifesciences LLP, which is her second entrepreneurial venture. It is a startup India recognized company. It is a research company endeavoring to build a value by developing the personal care, health and wellness as well as the pharmaceutical product. It has built up an array of patented and unique technologies and products in the topical and complex injectable sector, providing solutions for the unmet consumer and patient need. Prior to this, she was co-founder and director of Qpd Research Private Limited, which is India's pioneering and leading contract research and drug delivery company. As she was COO there for 17 years and instrumental in nurturing and building the business from scratch. Before starting her entrepreneurial journey, she worked for 15 years in the big pharma companies like Novartis, GSK and others in the various technical roles. Maharukh Madam is an active member of pharmaceutical fraternity and has been invited as a guest speaker in multiple industry conferences, academic institutes and companies. She is an active in the ICT Alumni Association and serves on the board of governors of the association in various roles such as Senior Vice President and Chair Distinguished Alumni Selection Committee. She is also on the committee of drafting the ICT startup policy. She is inventor of numerous patents and has also co-edited a book, Pharmaceutical Development. So, with this brief introduction, I would like to request Maharukh Madam to start with the session. Madam is going to talk on a very important topic of product development right from conception to commercialization. So welcome Madam and I request you to start with the session.
So good afternoon everyone. Can you hear me?
Yes.
Good afternoon. So thank you very much for the opportunity to be here today to share my experiences with everybody. And I'm really feeling very old after hearing all that. So forget about that. I'm still young. Okay. So I just thought that when we're talking of concept of commercialization in the pharma space in our sort of an industry, quality by design principles are very important, and we need to use those principles in our day-to-day working, and we do use them in the industry for the last 10 years plus because it has become mandatory. And it is not really something very difficult, it's very common sense and logical. But it's just a systematic approach to a commonsensical thing which we do by intuition. And I thought that it would be very good to discuss those theory and then maybe share some principles with all of you. Now theory, I've got quite a few presentations because these are part of a training series which I did for a European company 2 years ago. So I just put them all together and selected a few of those. I will go a little bit fast on this because this is the theory, so that we have more time to go through at least 2 examples if not one. And if you have any questions in between, please just raise your hand and stop me. It's fine, to cover it at the same time, and it will also help others if you have a question. So basically, we will first talk of what are the principal concepts in pharmaceutical product design and development, and why did the regulators worldwide and ICH feel the need to put these down. There is a big reason for it, which is related to our business and market. So basically, the problem is, if you look at the historical perspective, manufacturing has been plagued with huge drug shortages. Always, there have been a lot of drug shortages, lot of recalls, lot of supply issues due to manufacturing problems. You also know last year that lots of products, even Indian companies from US were recalled due to the nitrosamine found in the products and all. So there are a lot of recalls which lead to drug shortages. And the major cause is of course slow adoption of new technologies. We still use the machines which were invented 50 years ago. Our compression machine has not changed, right? Our granulation machine has not changed. In my lifetime, we went from a PLM to a rapid mixer granulator. That's it. There is no really much adoption because there is a big regulatory burden every time. So the industry also pushes back because the regulators need a lot of data and validation and all. So there is and therefore it is inability to predict the effect of scale-up. So there are a lot of scale-up problem failures. So products get developed in the lab and when they go to scale up, they don't perform, there are failures. A lot of batches are lost, yield is not there. So there are lots of issues like that. And why this comes? Because there is no complete understanding and inappropriate selection of the method, inability to analyze or understand reasons for the failures. And therefore, because of this, there has been it's actually caused the industry and the regulators a lot. Because when there's a problem, the regulator also has to go out and find out it's an issue of the safety of their or health of their subjects. And even the company suffers. So if you really look at it, these are all the root cause of drug shortages, and you can see lots of people have worked on it. So you have this, PDA has done some reports series, ISP has worked on it. These are all ISP is the Organization of Pharmaceutical Engineers, PDA is the Pharma Drug Association. So these are all industrial associations who have also gone and worked along with the regulators to find out, and this is what they found, There are short-term prevention strategies, practical focus. So these are all the problems which we find out, and then these were all the recommendations of these committees. And therefore, there has been a renewed focus on product quality. This is say around early 2000s when this movement started. And a lot of effort has gone into this. Basically, the idea is you have continuous product quality assurance with timely improvement. So the concept is that you develop a product, you scale up, you commercialize, doesn't mean that you stop improving the process or the product or anything. You don't have to be constant. The regulator has come back and said, be fairly flexible, keep on improving it, because that was the earlier thought process in the industry. Also, quality term was actually related to finished product, but more and more that the failures and recalls are because there is no control maybe on the input material or on the process or on the equipment performance. So there are lots of reasons for this. So just having a focus on the final quality will not help resolve these issues. And of course, quality has to be planned, and you have to design quality over a product life cycle. The full life cycle meaning development, scale up, commercial manufacture, and continual improvement. That is the full life cycle of the product development. And therefore, if you see all these various Guidelines have come up, and finally we have ended up with this Q8, which is our development guideline, Q9, which is the risk assessment guideline. What is it, Q9? It was much earlier. And then you have the Q10, which is the quality implementation. And then now you have Q11, which is for APIs, and you have Q12, which is for post-approval, post-manufacturing quality changes. How do you do them? How do you control them? When is the time you can do it on your own? When is the time you go to the regulator and things like that? And so you can see these are all the main ICH guidelines. So you have Q8, which is product development, Q9, Q10, and these work in tandem with each other. And then you have these other ones, there's one Q13 also now, which has come up, which is related to analytical method validation. So there are further improvements which keep on coming. And other initiatives has of course been the PAC. If you are aware of this process analytical technology, meaning online analysis of the batch, which helps you have a flexible process. For example, if you are blending a batch and if you have NIR technology to look at the blend analysis at various points, then you can have a flexible manufacturing process or mixing time. And then there's also a push towards continuous manufacturing technology, but I think we in India are very far from this. This is a very expensive technology, and when we cater only to generic products, it's very difficult to get into it. But this is also very interesting if you really get into it, if you get a chance to look at it. GIA has GEA, they have a full line, and it's very interesting to understand that. And of course, you have the quality metrics program, which is the latest one initiative. Last 5-6 years it's running. So if you look at Q8, this is the development guideline, where they talk that you need to have a systemic approach to design a quality product that begins with predetermined objectives and emphasizes product and process understanding, determination of a control strategy which is based on sound science and quality risk management. We'll talk more about it as we go on. So when you look at Qpd, what you're trying to do is you're trying to define and achieve meaningful quality attributes, and the attributes are linked to clinical efficacy and safety for the patient. The quality attribute is not for what your lab can test, it is for what the patient needs with respect to efficacy and safety. And that's how you go back, you design the quality into your product. And to achieve this in-house, you need to have an understanding so that you can reduce product variability. See, variability is a fact of life. You get paracetamol one day and next day there is going to be some variability. The idea is to control the variability in such a way that the output is fixed. You get a fixed quality output. And of course, I mean, these are all the other things you want to control the variations and inefficiencies. These all add up to a lot of industry issues and concerns. So the idea is to have a quality by design approach which will help you, design a product exactly as per the quality. So when you look at what are the elements, the first one is the target product profile or the quality target product profile. This identifies the CQAs or critical quality attributes of the drug product. So when you look at the therapeutic profile or the quality target therapeutic profile, there are many attributes. Some of them like color, size, shape, they are not critical. They are there for a marketing reason. But from the efficacy and safety purpose, they are not critical. But if you look at what is the related substances, degradation products, or when you look at the assay, when you look at the release, these are all critical. So you need to, for each product, and they are different. They are different for an injection, they are different for an eye drop. For each product, you need to define what exactly is critical and then control those in your development process. So your full focus of the development process is on defining how you can control the critical quality attributes so that you can control the efficacy and safety. And then you can also look at the critical material attributes. This is another element which is the attributes of the material. For example, if I am using HPMC as a binder, then the viscosity, the percentage which I use, both of them, the viscosity of the HPMC, maybe the solubility in the solvent, all these are critical for it to perform well as a binder. If I use it as a matrix forming agent, then the viscosity will be very important, but what could also be important is the particle size and flow and all, if it is a direct compression tablet. If it is a wet granulation tablet, then maybe only viscosity is important. So in each case, you have to understand what is critical in the material for the performance of the system. And then you have the critical process parameters, and then you have a risk assessment. Now, why do you do risk assessment? It's very simple. You do risk assessment because if you start listing down the variables, there will be umpteen number of variables. there'll be hundreds of variables, and you cannot keep on optimizing and studying each and every variable. So you select this variable is critical for this CQA, and whichever variables are critical, you study those and you control those. So the idea is that to control the variables which are critical to the product efficacy, and therefore that makes your development process very well defined and very contained. Otherwise you can keep on doing lots of experiments, but there's no meaning to that. And of course, these are all the other objectives. Oh, sorry, I went back. And then later on, after that, you come to a design of experiment. You can use an empirical or enhanced design of experiment to understand the critical and complexities. Basically, you can use DOE. DOE is a tool, but you can also do one-factor experiments like we normally do. And when you use this DOE, you use it if there are 2-3 variables which are causing an impact and you want to understand the impact of all the 3 variables together. For example, amount of binder, amount of disintegrant and lubricant. And if there is an interplay over there, then through the DOE and through an overlay plot, you can arrive at a design space. Also, what happens is that, what is the regulator saying? The regulator is saying that if you do all this experiment statistically and prove that you have a design space, for example, I have 2% of binder, but if I prove through the statistical experiments that I can have 1.5 to 2.5, then you can make those changes in post-approval situation without having to go back to the regulator, without having to file a request for re-examination of the data. So therefore, if you do this systematic approach, and if you have all this understanding of the product, you can have a flexible approach and your regulatory strategy becomes easier. So if you really look at it, what you are trying to do is you are trying to develop knowledge with respect to material processes, and then you are trying to establish the design space specifications and the controls. And when you say design phase, this is allowable, I just explained to you, a quantitable combination of materials which assure quality. Now, control strategy is very important because what you typically do is you have a risk assessment at the start of the project based on what you know about the materials and the product and your prior knowledge which you have of such products or materials. And then when you do the development, as you identify the variables and you study them and you fix the associated anticipated issues, your risk again assessment, when you do it, it comes down. And when your risk is at an acceptable level, that is when you design a control strategy and go for scale. That is how the process flows. And of course, this also means that there's a lot of knowledge which is generated in the organization, and you have to manage the knowledge, because what knowledge you get from one product, you can use for the other product. For example, if I am having a direct compression product with 1% of the active, and I have done a lot of work, then I do understand what are the criticality in the processing of a DC product, and I can use that in the next product. Maybe a very different API, different composition, but there is a certain base knowledge which you can use. And that is very important to be able to document that and use that. So, and then you have the ICH Q9, which is the risk management. So basically you are looking at the risk as a way to control the variability of the factors which are having a big impact on your CQA. And when you do the risk analysis, you know which ones you should study and which ones you should ignore or which are not important. Okay, and it's a systematic and sequential process, so you do it at different stages in the process, and it helps you make a scientific decision making. Now if you look at risk, so basically when you do risk management, you have these steps. You have a risk assessment, you assess the risk, analyze the probability, severity. When we see some examples, you'll be able to understand this. Then you have a risk control is a step, which analyzes whether the risk is within acceptable limits or plan a strategy to execute to minimize the risk. And then you do a review again after you have done some experiments, and then you generate it into a document which tells you that as of now, due to this experiment, the risk is minimized, it's low, it is at an acceptable level. Risk is never zero. Risk can never be zero. It's always low, medium or high, and you have to define those. So if you really look at the risk assessment, it's applied in development, in the control strategy also, and also in continual improvement. So every time it goes back, you have an identification, you reduce, it's accepted, you go back, you review the events. Once you commercialize it, you make 20-30 batches in a year, you review it, and again if you see that there is some failure, some batches are showing something erroneous on the borderline of failing, then you need to really go back, because obviously there is still some problem in the system. And then last you have the Q10, which is a pharmaceutical quality system. And this is a comprehensive quality management where it's complementary to Q8 and Q9, and it works as an interlink where the knowledge gained during product development and manufacture along with risk management can be implemented to ensure drug quality and also continuous improvement. Q10 pharmaceutical quality system stresses on continuous improvement. Nothing is static. You can keep on improving it. You will get new materials, new grades 10 years down the line. You might get APIs which are better, having better flow properties. So you keep on continuously improving the process and your product deliverables. And of course, these are the elements which are very part of the PQS system. And of course, most important is the managerial board is and responsibility. So quality is top down, not bottom up. The management board is responsible for quality and they are supposed to take responsibility for it. So this is the PQ pharmaceutical quality. It's talking of the same thing. Basically, this is the stage where it's an investigation product, then it becomes a GMP product. And then you have to manage all these responsibilities. You need to put in a CAPA management review, knowledge management, quality risk management. So these are all the elements, and this is what the guideline talks about how an organization should build this system. When you look at Q12, it is talking of product lifecycle management, and basically this is talking about how post-approval changes. How do you, CMC is chemistry, manufacturing and controls. So post-approval CMC changes, how do you do it in a more predictable and efficient manner, and you reduce regulatory submissions. So this is what this guideline is talking about, and it's recently been issued, the final one. So this is also very, very helpful because many a times, in the past you have to go back to the regulator and say, I've changed this process, I've changed this, and wait for. So you have to generate a lot of data about the change, apply it, tell them, ask them to review it, and then only you could make the change. But now with this sort of a system in place, there are certain types of changes which you can make on your own, and during the annual quality review, you submit it to the FDA. You don't have to really ask permission. You can make the change based on your own knowledge. So this is a very important slide. I don't think you can say. But basically, what happens is that. Okay. So what happens is that when you are in Q8 is development. Okay. This is the guideline related to product development. And here the role of Q8 is maximum over here. And as you get into commercial manufacturing, the application of the Q8 requirements go down because by that time your target profile, your CMAs, your CQAs all are defined, your process controls, everything is defined. Q9 is risk management, which actually continues throughout the life cycle of the product. Q10 is quality management, again, which continues throughout the life cycle of the product. And Q12 is your post-approval. So the impact of this starts after you start commercial manufacture over here. But the key over here is that as your knowledge, as you go from development to tech transfer to commercial manufacture and product discontinuation, as your knowledge increases, your risk decreases. So you are having continual improvement because you are increasing knowledge on your product and you are reducing the risk. So quickly we'll just go through some of the concepts of what is a Qpd concept, and basically it's a very simple explanation. Where is the industry today and what are the outcomes? So Qpd is a very different way of thinking, that's all it is. It's not just in processes, understanding and implementation, but also people and mindset. It's a mindset, an ability to understand what's happening to the product, to the process, and then resolve the problem instead of trying to do any short-term solutions. So basically what it says is your input is your tangible process, people, quality, mindset. These are all the, tangible is the process, but rest everything is intangible. And when this input is there, the output, you get an output depending on what is the input which goes into it. The output is totally dependent on the tangible and the intangible. So quality means doing right even when no one is looking. And basically it's a systematic approach with predefined objectives, product and process understanding is the move point around it, process control, sound scientific understanding and statistics, and quality risk management. These are all the elements. So if you really understand it, when you have a fixed process with variable input, typically you will get variable output. That is what was happening. Now if you have better understanding, control strategy, and you can reduce the variation, then you can have a variable input, but you can have a flexible process and still get a fixed output. This is what Qpd is all about in a nutshell. It's all about controlling the variation by understanding the product, the process, the equipment, the mindset, everything. And when you look at risk assessment, you are leveraging your product knowledge. Okay, so what we know, what we don't know, what we don't understand, but this is the danger part. When we don't know something which is not obvious, sometimes we don't know many times what is going wrong, or we cannot anticipate, we cannot think, and this is when you get unexpected failures. So if you have a structured approach, you will typically not have this, because you will, whatever is not obvious also, you will know if you do a systematic study. And so basically we do everything intuitively, but these are the structured tools which are used for risk management. I'll show some examples of some of this. I think that's okay. Everybody knows variation is a constant in life. And we all know this, that, as the risk and variation increases, as risk increases, variation also increases. Similarly, as variation increases, quality goes down, customer satisfaction goes down, and your cost escalates. So therefore, understanding and reducing variation leads to robustness, and that is Qpd. This is Qpd in a nutshell. And it's same thing with process understanding also. If you look at what are the common causes and special causes, there are many causes. Of course, human diurnal variability, natural material variability. This human element, especially, night shift working and all, it's very well known and very well documented that most of the errors happen at that time. And you can't always control everything. But if you understand things, then you can maybe minimize and eliminate these sort of OOS. OOS is out of specification or out of trend, OOT. These sort of results, because the moment you get into that, then you get into a big QA investigation of why that happened in a manufacturing setup. And of course, you have special cause variation. So, there are lots of causes, but most causes of OOS and OOT, you need to understand the root cause and eliminate, otherwise it will keep on happening again and again. And doing a root cause analysis for an out of specification of a batch or an out of trend result is very, very difficult sometimes to get to the root cause. And then typically what happens is, many a times we will say analyst error. Analysts go oppositely trained to know. But we don't know what's the root cause, and that doesn't solve the problem. And I cannot every 6 months say there is a failure on a batch and it is analyst error or manufacturing error. Then something is wrong in your system if your people don't understand what they have to do, or maybe there is something else which is wrong which you are not able to pinpoint. And that's where this is very important to understand. And that's why the management has to review the quality. They should have a monthly review and they will understand, and somebody should pick up that we are constantly having analyst error. What is wrong in our QC lab? Or we are constantly having some manufacturing control going wrong. What's wrong in our manufacturing? Somebody has to question that and rectify that. And this is the thing, that when you don't have control, you get into rework. And when you get into rework, then it really creates a lot of issues, because the efficiency goes down. You have to do so much investigation. When you have the next audit, again you have so many problems to understand and explain that to the regulator. So it creates a lot of backlog of work, which you don't want. So basically in the past we are reactive. Overworking ho gaya, we will say analyst error, retrain the analyst, forget about it. Close that investigation for now and go ahead. But that doesn't help, because it happens again and again. And then in the present we are proactive, but we need to go to the predictive state. That's all that we are saying. We all know that when you are working hard, then your performance will come down with time, effort will go up, capability will go down. But if you are working smarter, you will have actually better performance, less effort and more capability. So Qpd is done, the problem with the industry today is that the Qpd is doing with what is the regulator wanting. Regulator को ये चाहिए मैं दे देती हूँ वो आ गया मेरा PDF में आ गया चलो file approve हो जाएगा, but that is not what Qpd is, because your customer is a patient, not the FDA or not your EMA. So therefore, this is very important to focus on patient needs. And also perceptions, we have to comply to Qpd or fixed requirements, or actually ऐसे कुछ compliance ऐसे नहीं है. You are supposed to apply that as an approach. You are supposed to use that to generate knowledge of your product. So again, many people feel a robust Kappa is Qpd, but it is not Qpd. If you have corrective and preventive action, so the problem हो गया मेरे manufacturing में, if I correct it and prevent it in the future, it is Qpd. It is not Qpd. It is just like a chigar patti, It's like a band-aid. It's not really a solution to the problem. So you cannot use this. Anyway, these are the problem statements. And then grey areas is, of course, the analytical method development also should be done as per quality by design principles. And recently this draft actually has just come out 3 months ago. And so even in method development, when you're working in the analytical space, you need to apply this quality by design criteria and then develop a method. So it's the same principle basically. Even sampling, where is the sampling? Sampling is a big problem, and there are many issues related to sampling. If you read any of there are many review articles also on this. So sampling can actually lead to erroneous products getting out into the market and create a lot of problems. So there is sampling of, we do unit dose sampling and things like that, but it's always a problem because unless your process is very thorough, you will always wonder whether the sampling is really representative of the batch and does it tell me that each and every unit is safe and efficacious. So basically even process validation we have the same thing. So we have the new process validation guideline which came out a few years ago, where again they are talking of a life cycle approach. Earlier what we used to do, 3 batches, validate kiya, khatam ho gaya, wahi process chal raha hai. But it doesn't end at that because there is always a requalification and a re-look at the process which needs to be taken periodically. So if you look at it, basically it's a life cycle approach. You have to define, measure, analyze, improve, control and restart. This is what we are talking about, and it cannot happen without a team. Qpd cannot happen without a team. Risk assessment cannot happen without a team. Because when you do risk assessment, you need people who are into development, who understand the product process. You need somebody who is in manufacturing. You need the engineers over there who understand the equipment, and then only you can do a proper risk evaluation. Equipment maintenance, again, this is another big problem, and there are many ways to do this, but this is a lacuna which is always there. So basically we are generating lots and lots of data instead of having a lean manufacturing, because we keep on trying to justify the changes which we keep on making instead of trying to build the knowledge and have a robust system so that we can predict what is going to happen and take care of it. That is so basically from an unstructured we want to go to a structured sort of a system, and that's what the Qpd approach is all about. And so you define the design, you measure it, you analyze, you improve and you control, and this keeps on going. This is what is a continuous improvement loop. So with time, as your quality and understanding improve, variability will come down and this will improve the quality and understanding of the product. Of course, I find this very good. It's neither the strongest nor the most intelligent, but those that are more responsive to change that survive. So you have to change with the times, and we have to modify our thinking and the way we work. I hope I'm not going too fast, but I'm just trying to save time for something else. Is this too boring? Very soon we jump into some examples. Yeah. So when we look at Qpd, we have got the elements of the Qpd, we discussed about it briefly. Basically it's about planning quality and controlling it and improving it. Now the first one is the target product profile of the Qtpp. So you prescribe what should be there, what should be the target that the product should be. This is with respect to physical properties, chemical properties, pharmacokinetic, biological properties, and then you have the other things like packaging, stability, microbial contamination, and also you define it. And then so it's an extension basically of, and TPP is typically what happens is when you are dealing with new molecules and all, you will start with a target product profile because you don't know what is the quality expected. Suppose you have a new molecule or a new, you're trying a new delivery system. So you have something theoretical, but you really cannot define the quality. So you start with a target, you start your work, the moment you do your animal studies, you understand the safety, and then when you go to phase 1, that is the stage when you can define the quality required before you go to human studies. So it's different for generic products and for new drugs. But finally at the end of it, you are actually trying to concentrate on the desired therapeutic effect that you want. And it is also safety and efficacy. In all regulatory requirements, regulator is only asking about safety, efficacy and quality. These are the 3 things. Everything that you do in the pharma industry is related to these 3. So whatever you whatever data you generate, if you can relate to either one of these elements, that means you are on the right track. It's very important to keep that always at the back of your mind. So, I mean, for a new product, you start at the start, you define it, and then you keep on improving it. So you may define something is being very critical at the start of the product, but as you study it, you may find that it's not very critical. Even without controlling that, you are able to get what you want. So sometimes these things also can happen. So you need to look at the data, look at the science, see what you want to give to the patient, and accordingly change your systems. Now the Qtpp, it forms the basis of design because everything is going to what you're going to do further is going to depend on this. So it starts with what dosage form, what container closure system, what is the moiety, what sort of PK you want, what is the quality, like sometimes you may need sterility, purity, stability, drug release. So for the intended marketed product, what is it that you need to control? And typically if you look at it, see this is like for an IR tablet. So, when you're looking at a target of 30 milligram, the corresponding CQA will be identity, assay, uniformity. This will be critical so that this dose is given. Physical properties are there, but these are not really CQAs in that sense. These are more to control the consistency of the product. Then you have the particle size or something like that, which could be very critical for the bioperformance. So, acceptable PSD of the API and drug release can become your CQA. When you look at safety, then the degradation or impurities become very important. Chemical and drug product stability, about the shelf life and the appropriate packing. So this is how these two are, they get related side by side. I think I'm not going to go into this, we will do the example. So and what happens with the TPPs, they evolve during development and with a better understanding. So this is the product profile, you begin with the end in mind and you define what you want. Then you look at the CQAs, you decide which are the CQAs. So they link the product quality with the desired therapeutic performance. Like I told you, tablet weight and tablet size will not be a CQA. What will be a CQA will be the assay and the content uniformity. Similarly, so it's what is a CQA? It is defined as a physical, chemical, biological or characteristic that should be within an appropriate limit, range or distribution to ensure the desired product quality or the therapeutic performance. And it can be different for different products. Although they are deemed important, it is important to understand that how they can potentially affect the patient therapeutic outcome. So when you are looking at the acceptance criteria, you don't look at what your method can determine. You have to look at what is required for the therapeutic performance. So if you have a method which cannot quantify a degradant below 0.5% and that required for safety is below 0.2%, then definitely it's an inappropriate method. So you need to correlate even your measurement tools along with what is required for the product performance in the therapeutic situation. And therefore what we are looking at is performance-based quality specifications. For example, this you see size of beads and drug products labeled for, there is a specific guidance on this. So you have lansoprazole beads, enteric coated, which people, I think, empty out on their food, sprinkle it on their food. And for that, there is a particle size requirement and there is an FDA guidance on it. Because this is a very critical attribute for the clinical performance of the product. And therefore, there is a guidance for it. So this is again a very unusual need, but that's required for the therapeutic performance and therefore, this is coming to be. So then, I think we already talked about this. Sorry. Then you come to risk assessment. I said like they are very different tools and you do the risk assessment so that you know what is the high risk and you bring it down. Then you come to the, so you have the critical material attributes, as we said, a material attribute which is critical for the performance of the system. And when a realistic change can lead to a significant impact, that is the attribute which is very important. Risk assessment is a very valuable tool in prioritizing those material attributes. So typically what you do when you start a product, you look at the Qtpp, you define which are the CQAs. Then you define what are the material attributes of the API which will have an impact on the CQA. For example, if it's a very low dose drug and I have a direct commercial tablet, I will say that the particle size of the API is very critical so that I get a certain content uniformity. And that is critical because the efficacy is related to it. Or if you have a product which is having a very high degradation rate, then maybe water content in the API is very critical to prevent the lability of the molecule. So for each product, depending on the chemistry and the way it behaves, you have to define what is a critical material attribute and how you can control it. Similarly, process parameters are also need to be controlled, and whichever is critical, you need to, and significant impact, you define that as the CPP and you study that. And to study that, you have a risk assessment. So you define what is critical and you study with that. Prior knowledge is very important, this is very, very important because we don't have to reinvent the wheel every time. So it can come from literature, it can come from knowledge of other products, it can come from knowledge of the team members who have done similar things, it can come from the knowledge of the engineering team who understands the equipment. So this prior knowledge is very important and this should be used, you know. So, this is very difficult to define, but if you really start thinking as a common sense, you will know that you already know. Magnesium steroid, if I mix beyond 10 minutes, I am going to have an impact on dating. I know it. Why do I have to do it to prove it? There is no sense in that. So this is a type of knowledge which everybody knows. When you are working in the system for a few years, you know. This you cannot do because it has this impact. Now you don't have to do an experiment to prove that, which you already know. And that is the point where the prior knowledge needs to be documented, and you document it when you do the risk assessment. Again, I will show it to you in an example. So, and then you come to the design space, like I told you, the space within which it operates well. And then there are different ways you do these contour plots, so you come to the design space. So basically, parameter and parameter 1 and 2, when you control them in this space, you are going to get something which is passing your predetermined specifications. And therefore, this gives you the flexibility of changing this without having to really do any work. If you have done this sort of experimentation and you have this understanding and data, you can make changes without having to go back to the regulator or without having any concern on the product performance. Similarly, you have the overlay of the two parameters and then you can have the combined design space. So, basically, for design space, you need to do DOE because you are looking at 2 or 3 variables together. Typically, you do the center point method, which is the easiest one and very often used. And then you have a control strategy, which is based on different elements. So, basically, you have the understanding of the ingredients formulation process, you do risk assessment, you understand the impact on the critical quality, CP and CM, and then you have the acceptance ranges and you come to the control strategy. This is the full process which you follow. And when you have this sort of information with you, even if you have some unanticipated problems, you have enough understanding to be able to sort it out when you get it. And continual improvement is a fact of life, and we have to do this so that we can keep our businesses always going well. Okay. So, now, when you look at Qpd, applying Qpd risk assessment and quality risk, it's an iterative process. It's not a constant process that I do 10 experiments and it gets over. It's an iterative process which has to, it goes up, you go back, do risk assessment. If it is okay, you go up. If not, you go up again. So it's an iteration which keeps on happening, 1, 2, 3, and then you reach your stage. And it's very important for us to understand that. I think we've already talked about some of these things. Process design is again very, very important and very critical. And also in process design, what happens is that you have scale dependent and scale independent parameters. For example, if you are looking at compression, whether you compress on a 8 station or 16 station or 48 station, the basic process doesn't change much. It is only the machine parameters of the speed changes. But when I am looking at fluid bed drying, then a 1 kilo and a 10 kilo and a 100 kilo is going to be a very different thing. So what happens in development is we have limited equipment, and those which are process independent parameters we can study in depth. So that we can straight away apply when we go to scale up. But for the process dependent, we study to understand the cause-effect relationship and have that information ready. So, but we need to do a lot of study or qualification even at the scale-up level, because at that equipment level, if it is a process-dependent or scale-dependent parameter, we need to also consider doing a qualification or a study at that level. Now, how you do it very optimally, because nobody wants to go to a 100,000 tablet or a 100-liter batch and do so many batches. It's very expensive. So that is where you need to understand as much as possible and design the experiment in a very efficient manner. So basically, process and product design development, you cannot separate. They are all go together hand in hand. And a process is supposed to be well understood when you understand all the critical process variables, and your control enables the management of the variability. And therefore, sometimes you need to do something even at the scale-up level, because the equipment is different, way it behaves is different. When you have a small mass and large mass, again the behavior is very different. When you are granulating, or even when you have a suspension, when you have 100 liters and 5,000 liters, it's going to behave very differently. So that's what you need to do at that scale. I just want to show you this. Okay.
It's okay. It's okay, you can see. That is okay.
I'll read out.
You can see.
So basically, how it goes, how the full process goes. You first define what is your TPP or your quality target profile. Then you define your process formulation and manufacturing process. You cannot really do a risk assessment unless you have that, okay, I'm going to make ibuprofen tablets 400 milligrams with a direct compression process. And I'm going to add lactose and starch and HPC or whatever as a binder and then a disintegrant and this. So unless you have a defined product and process, you can't really go ahead and do your risk assessment or do your definition of the CQAs. So you really need to have this in place. And therefore, sometimes, you have in new products, like I said, you have a target profile which says, okay, I want to have an anti-TB drug which is giving me this sort of efficacy or whatever. But unless, and safety. And unless you do the preclinical work, you cannot really come back to a Qtpp. So, but in a generic space, typically it's very easy to do that once you have defined this. And then once you know what is the formulation and the process, then you can define what are the critical quality attributes for the drug product. What is critical for that drug product? So I know I'm going to make ibuprofen tablets by wet granulation process. What will be critical for that particular product is very easy to define. If the formulation in the process is not defined, it's very difficult to define because it will not really be accurate. Once you have defined that, you look at the materials. So you define what are the critical material attributes of the drug substance, excipients and in-process materials. Okay, all related to the CQA. So what in the material attributes of the API or of the excipients which are used, say MCC, lactose, particle size, water content, blood density, or even the in-process material, the granulation at different stages. What about these stages is very critical for the achieving the CQAs. And then you apply your prior knowledge. Using prior knowledge, you do a risk assessment. Okay. When I do a risk assessment, I know which are the variables in the composition which I need to study. For example, level of binder, level of MCC, disintegrant level, particle size of the API, So you do experiments either empirical or DOE based, and when you do the experiments you get the data, when you update the risk assessment. Once I get the data, I know that particle size of the ibuprofen is very critical. When I do the experiments and I come to the understanding that if I have a particle size of 10 to 50 micron, I'm going to get a product which is complying to my in vitro specs, or it's complying to my compressibility and everything. If everything is fine, I will fix that as a range. That means that risk has been minimized, it's come to zero. So you update that risk, and when you update the risk, then if it is okay, then it goes into a acceptable level. Similarly for the process, you do a risk assessment with respect to the critical process parameters. So in development, you will only of course look at all the parameters, but you will concentrate more on the scale independent parameters. And then what you do is you do again experimentation to understand the process, to see what is critical, how you can control it, in which range is going to give you the product of your desired Qpd. And then you do an update. Now when you have defined this, what happens is that you have defined the quality process parameters and the materials, and you come here. In case the risk is not acceptable, it goes back. It can go back either here, maybe there's something wrong in your proposed formulation itself, maybe the binder you selected is not correct for the product, or it can go back to your material attributes, so it can go back again to experimentation. Similarly over here. If it is yes, then you will go further. So you've got enough formulation understanding, process understanding. When you go further, then you define the design space and control strategy. So here is the place where you have opportunity for parametric or RTR testing. RTR testing is real-time release testing. When you use packed systems, or if you have any such facilities, then you can do this sort of a controlled release. You come back to the original, then you come to the overall control strategy and batch release strategy, and then you go into commercial manufacture. Again, you are supposed to have a risk review, and then again it goes back into the loop. So if you look at it, you are going back so many times. You are going back more times than you are going down. So this is an iterative process. It is not a constant process. This is very important to understand. And even after commercial manufacture, you can still keep on improving. So this is very important to understand this, that it is actually not a one-time process, and therefore you start development in 2022. By that, oh sorry, what happened? You start development in 2022. By the time you come here, it is 2024. By the time you realize something in 2026. Now, if you don't have documentation of this knowledge, you will be reinventing the wheel again and again and again and again. So knowledge management is the key to any organization. How you manage your data, your knowledge, how you see that it's easily available, retrievable to the teams, and it's kept in a proper. So the QA department has a big role to play in containing this knowledge in a retrievable and easy to find way. This is very important. Otherwise, you will keep on doing things again and again, and you will lose out on some very key parameters. So anyway, these are all. I am going to skip this. Just come to this slide. So in a nutshell, what is the ultimate goal? Gain an in-depth understanding, reduce product variability and defects, and enhance patient efficacy and safety. This is all that you are trying to do. By doing all these things, round and round, round and round, finally that is the goal which you are trying to achieve in our development process. I think I am going to straight away jump into this. So this is a chapter from my book on product development, which I co-edited with Vandana, Amaterasu and Dr. John D'Souza. So I'm just trying to show you a few examples from this.
Madam, that is a very good book, but the soft copy of it is not available.
You don't have a hard copy?
We have only 2 copies here. I mean, we can purchase, but if soft copy is available, then
Then of course Taylor and Francis to do that. Okay, put this on.
it's a switch.
It's on? Is it on now?
Yeah.
Okay, fine. So I just quickly take you through this, that how do you build an effective product development strategy, and we'll have some examples in this quickly. So basically the need for developing a robust process to drive the development. It's very important to have a very robust process, because your product development is only as good as your strategy. The amount of effort you put in reading literature, collecting information, understanding what you're trying to do,
understanding if there are any comparator products. The more effort you put into it, the more tight is your strategy, and you will hit it right the first time. So we don't really want to do trial and error. It should be a very predetermined, targeted strategy and targeted experimentation. That will give you the best results in a proper way. So basically, it's of course very important, the strategy is very important, and you need contribution from a lot of stakeholders. So this is very important in any team, you know. Development people think they are pushing, they know everything. ऐसे नहीं होता है. Okay, there is so much input which you can get from your colleagues who are working on the shop floor every day, or your engineering people, even also people in the lab who are analyzing. They will tell you, ये ऐसा फुलाना tablet madam इसका dissolution fail होगा. Without doing analysis, the analyst knows which batch will have a lower dissolution or a faster dissolution. That's a lot of knowledge in there, and you need to get it to the table to really use it. So therefore, you really need to form a team to finalize a strategy. And it's again, the strategy is not static, it's dynamic. Okay, I do a strategy today, I do experiments. 3 months later, when I review everything, I might say that, "Arey yaar, this strategy galat hai. This is not working. We are not getting the required dissolution, or we are not getting the required bioequivalence or stability or whatever. Go back to the drawing board." So that can also happen. At the same time, you may learn something which may tell you that, okay, if I just tweak it a little bit, I can get a better release or a better safety profile or whatever. So therefore, we made a strategy, so we have to stick to the strategy. ऐसे नहीं है. Finally, what we are trying to do, our goalpost is not to stick to the strategy. Our goalpost is to reach that product which, you know, complies or satisfies all my Qtpp. And I get the therapeutic target profile exactly like what I want. So therefore, it is a very dynamic thing. I'm not going to show you this, that's okay. So you need a team of subject matter experts for discussion and brainstorming session. And what is very important is you have to have a document which systematically evaluates available prior art understanding, product requirements from the patient's perspective. And based on that, you define the Qtpp and the CQA. So very important to do this, and typically a strategy document should be approved by the quality and regulatory experts. Because I may make a strategy saying I'm going to do something, but if from the regulatory perspective it's not acceptable. Yeah.
Actually, there are so many Qpd models they have shown for all these pharmaceutical products.
Yeah.
We have that Qtpp, CQA, CMA, with examples of tablet, capsule, parenteral injections and all are there. The main is that suppose if you want to develop a biological product,
Yeah.
so it would be better, madam. So what kind of model?
See, the concept doesn't change. You understand the concept and you apply it. So if I have a biological product, what am I going to look at? If it's a protein, I'm going to look at the primary structure, secondary and tertiary structure. I'm going to look at methods to evaluate them and see how I can control that structure. What is in the structure is critical for the product to give me the required therapeutic efficacy. So it is the same concept. When you have the what they call it, that the cleaning process रहता है ना end में after fermentation.
Yeah.
When you have the back end process where you do the purification, and there what are you going to use? Materials. What impact the quality of those materials are going to have on that process? In the fermentation step, typically you will have a fermentation step. What are the medium and the other things which you add over there, or the cell lines which you use? What are the critical attributes of those which are going to impact the processing? So you have to use the same concept. I have not looked at biologicals, I'm sure something is available. But again, you have the maps and then you have...
So even the regulatory guidelines are also not so clear for the this biological products. Vaccine, suppose you want to go for a vaccine. Okay, so in that case, yes.
WHO का guidelines है ना? WHO का guidelines होगा ही होगा vaccine का. I have not seen them basically because I have never needed to. But I am sure there will be guidelines. WHO you can look at. But even for biologics, I mean there is a BLA guidelines are there, no? FDA का सब biological license application का. There are lots of guidelines.
Yeah.
What they look at, what not, when you develop, what are the parameters to look at. वो है, guidelines are there. Some basic guidelines. See, what does a guideline tell you? A guideline tells you what is the regulator looking at when he's looking at your file.
I'm talking about madam the product development based on QBD.
Yeah, but product development based on QBD also, it is, the concept is same. You define your target profile which is linked to the patient therapeutic performance. Then you define the quality attributes of the product which are linked to that, safety, efficacy, right? And then you go further into the materials. Now here in this, in biological, the material will be, you start with your cell, which is going to give, you're going to ferment and give you the cell or whatever is your medium. Yeast cell है कि oyster cell, क्या है, hamster का cell होता है या क्या होता है, what is the medium? And what is the medium you're going to use? So you will have to go into those and evaluate what impact it has. So people who are skilled in that, who have done that, will know कि when I take a hamster cell, which cell line will give me what sort of thing, and for which cell line what sort of medium I need, what sort of temperature conditions I need. So I mean, people skilled in the art will know. But the concept is same. You understand? The concept is same. It doesn't change. For example, we are developing a once a month injection for Alzheimer's. Now, I mean, there is no product like that, but we will go from the pharmaceutical principles and say that when I want to develop the product, first of all, it is an injection. I cannot have more than 3 ml. I cannot inject more than 3 ml. बराबर है, वो मेरा एक criteria हो गया, ठीक है. Then I will say I should have contained so much. There is a dose calculation. When instead of once a day, if I want to give once in 28 days, there is a dose calculation. So this is the theoretical dose which will have to be further studied. When I look at PK, I will say, okay, when I am giving oral, this is my Cmin Cmax. I should get something similar to this at steady state. Okay, in a tablet, the steady state is 5 days. In an injection of 28 days, the steady state may be 5 cycles, 6 cycles. I have to find out. I don't know how many cycles it will take. But from our biopharmaceutical calculation, we know it should happen in 3 cycles. So after 3 cycles, this is my steady state Cmin C max should be within this, or C average should be this at steady state, and it should compare with the tablet. Why? Because the tablet is known to have efficacy. It's known to give the anti-Alzheimer's effect. I am trying to develop once a month, so it should have something similar to that. Otherwise, I will not be able to show efficacy, right? And then you come to your container closure system. It's a sterile product, so it has to be sterile. It will have to be in a container closure system which will keep it stable. It might need nitrogenation because it's an injection, you know. So therefore you come, and then I will have to do the endotoxin testing. So that's how you will come to the Qtpp. So some are linked directly to the performance, others are linked to the safety of the dosage form. It has to be sterile, it has to be leak proof, it should not have particles. This is all linked to the safety. You understand? So the concept is same. You don't need an example. You go by the concept. If you read that Q8 very carefully ना, तुम 4 बार पढ़ोगे ना तो आपको समझ जाएगा. And you read Q8, Q9, Q10, read it in conjunction. They are all correlating. Q8 will talk about how Q9 can be used, Q9, Q8, Q10. They are all integrated together. All the concepts have to be used together. You can't only use Q8, product development by Qpd, only risk assessment or only quality management and life cycle management. You have to look at them in tandem with each other and use all the concepts together. I hope I answered your question. I'll try to find out if there are some guidelines that we can do. Okay, so therefore prior knowledge we talked about it, it's very important. So therefore if we come to this ibuprofen say example, so we are looking at what are the physicochemical properties of the drug. We try to put them down, you know, they have a melting point, no melting point, again a concern for processing. And what is the pK, what is, so these we define all this. It's very important because all of these have an impact somehow or the other on the performance of the system. And then, okay, what is the type of class it is, you define the Qtpp. So when you go to, when can a Qtpp be defined? That's very important. So for a new chemical entity, basically it is supposed to be defined after you have animal and clinical phase 1 clinical data. So that you know what is the dose that which is safe and which will go into phase 2. So before you go into phase 2, typically in phase 2 you do dose ranging. So before you go there, you know what is the safe maximum tolerated dose in humans, and then based on that, you also have all the animal data relating to safety. you know what the impurities are, stability are. At that stage, you define the Qtp. There's no point in defining it first because it will keep on changing. Your formulation will also keep on changing in the animal stage and all. Typically, animal stage में तो you don't even have a formulation. Sometimes you just give the drug as such. गवाच करके देते हैं. So when you come to phase 1 is when you have the first human experience, and then putting both together, you can arrive at a target product profile or therapeutic profile. And then when it's a new dosage form, like I said, once a month injection instead of once a day tablet, then you define it as soon as you conceptualize, because you need to have some target in mind. You already know the molecule. Okay, I know the molecule, I know how it behaves, I know its absorption, PK, I know the elimination route, I know what is a T-half. So considering all that, I can define a TPP. For a generic product, I can do it very easily, but even for a 505(b)(2) or a new dosage form, you can do it very easily because you know what is the efficacy and safety of the current product with that particular drug. So you can use all that information and you can define a QTP. So if you look at film-coated tablets, so just quickly we'll go through this. If you look at the Qtpp for film-coated tablets of ibuprofen, then you will say it's an immediate release tablet, oral, analgesic. This is the dose. It is equivalent to the marketed product. And I mean, this is the justification and the reference. Now when you go forward, you have the tablet weights again defined. Then you have, again, these are all related to manufacturability, length, breadth, hardness, thickness. You may have this, if you're in a generic product, you already have the comparative product. So it's very easy to define this because that is the strategy. You have a strategy with related to, if you have different strengths, then you have to work out a strategy related to biostudy. How many biostudies you are going to do, and based on that you will have the dose weight proportionality. So that is very well defined and there are very good guidelines for that. So you don't have much of leeway over there because you want to be as close as possible to the marketed product from the marketing point of view. Disintegration time, triability, these are typical pharmacopoeia limits.
Madam,
Yeah.
if we are changing the dosage form, Qtpp can be applied.
Yeah.
For ibuprofen being a tablet, if I produce an ibuprofen nano suspension, I can
Of course you
apply
can
that
apply. Why not? I just give you an example of once a day to once a month injections. Qtpp you can apply to anything. As long as, you know, it's easy to do it for a known molecule.
Yes, for the phone.
Yeah. Okay. Then the assay, uniformity, related substances. Again, I mean, these are targeted for clinical effectiveness. Related substances, typically degradants. Release in the media, water content, residual solvent, microbial testing. And then you have the biological, what should be the, you know, Cmax, Tmax, AUC. I mean, this is a generic product, so definitely we need to match 8125 with the marketed product. And then you have the packaging related, storage conditions, container closure, where you may specify that I want a stability of at least 24 months. You may have a marketing requirement which will say I want at least a 36 month stability. Then you put that in because you have to develop the product and the pack in such a way that you get that stability, considering the chemistry of that molecule. Administration, alternate methods of administration. So what happens is that this is your QTP. Now when you define the formulation and process, like in this example, if you see there are two processes. After a lot of study, you come to two processes. So this is the formulation which has been defined or sort of, and then you define the CQA. So what you do over here when you define the CQA is you look at which is the CQA which is critical, which is not critical, meaning they will anyway be, it's not really totally affected directly to the efficacy and the safety. Those are not CQAs. They are a quality attribute, but they are not critical quality attributes. So you will not study the effect of the variation on those during development. For example, microbial limit. For tablets, we will always have less than 100 CFU and less than 1000 and not more than whatever 10 yeast and fungi. That's a standard pharmacopoeia limit for a tablet dosage form. Now that is coming from the input material and the processing control of the area during control and how you are controlling the cleanliness. That has got nothing to do with my formulation or my process. So that is a quality attribute, but it's not a critical quality attribute. And I will not then study it during development. You understand? That's how it goes. So LOD or water content is critical because it can have an impact on the stability. So it is very critical. Similarly, appearance is not critical. Sorry. By density, again not critical, particle size may not be critical or medium critical. Blend uniformity of ibuprofen is essential. You need to, but because the drug content is more than 50%, uncertainty is not very high. Hence, criticality is medium. So like this, you have to give justify for everyone why, and this is common means. This is common knowledge. We know what happens if it is. If I have less than 1%, I know it's going to be very critical because getting that content uniformity even in a very granulated product sometimes can be a challenge. So therefore, you apply whatever is your known knowledge and you define the criticality. This is for the next trend.
Ma'am, how do we differentiate between the low and the medium critical?
Yeah, so therefore, if you see every year, I have given you a sort of a justification. So whatever is impacting the efficacy or the safety, and whatever is controlled by your formulation or your process, that is critical. Okay. For example, size, diameter of 10 mm, that is controlled by a punch size. It is not controlled by my composition or this, so it is not critical. But it is a quality attribute because if I define 10 mm size, the size should be 10 mm. That's a quality attribute, but it's not critical. But if I say degradation products should be less than 2%, it is very critical. If it's more than 2%, there's a safety risk. Or drug release is very critical, assay is very critical. Whatever is controlled by your formulation and your process, that is critical. That's what you will study in development. What is not controlled, which is controlled by your, for example, sterility. In the case of sterility of a sterile product, that is controlled by the cleanliness of your area, which class area you have, and whether you are doing and your sterilization process. Okay, so your process of sterilization is very critical, but the sterility test as a quality is not critical. And so, therefore, I think. So now what we are doing after this is we are doing the criticality of the core tablet. So see this, if you look at it, these are the core tablet parameters. Now average weight is very critical because the dose is dependent on that, assay is dependent on that, right? Size is not critical at all. It is just defined by the punches, right? Uniformity of dosage form, again it's medium. Why it is medium? Because it's more than 50%. Because it is more than 50%, the chances of happening is very less and it's a very granulated process. If it was a DC process, it would still be critical. But there is a medium criticality, so you will study weight variation again, it has an impact on the dose. So it's medium criticality. Dissolution is highly critical because you may not get bioavailability. Dissolution doesn't match. Similarly, for the potent tablets, what is critical, what is not critical. Okay, I say dissolution related substances. Residual solvents again is not critical because if I am not adding any solvents in the manufacturing process, then where will the residual solvent come? They will come from the API residual solvent or from the excipient residual solvent, which are anyway controlled at the raw material stage. It's not coming from my process. So it's not critical from that perspective. Water is critical if I'm doing granulation, and water always plays a role in many other things. Again, microbial enumeration test. This is a standard pharmacobial test and we are doing it. Now it is non-compliance will impact patient's safety, definitely. So it's a quality attribute. But material proposed to be used have low microbial load, water content is controlled, which ensures low risk of microbial growth. Therefore, this is considered as low. And it will not be discussed in detail during the development process. That's what we say normally. We will look at it when we go to manufacturing. When I am developing, I will not concentrate on the microbial limit test because it's not controlled by my process or my
justification that you're giving.
Yeah.
Like it's what if that justification is not available? What this is from literature, whatever it
This is not literature.
okay.
This is not literature.
No, justification.
This is not from literature. This is basic knowledge which we have put into justification.
Okay.
You have to write it yourself.
Right. But that becomes qualitative thing. Now, if what if I have to prove it? Okay.
It's not qualitative.
We are not supposed to prove it statistically that it is low, medium, or when we are comparing, formulating.
I am defining a target profile.
Yes.
I want to achieve this in the product.
Yes.
In order to achieve this in the product, what are the critical quality attributes
Yes.
of the API or of the granules or of the tablets? That is what I am defining. Okay. Now, when I am defining the quality attribute, these are all the quality attributes which are well defined. Pharmacopoeia mein jao to, if you
Yes.
go to a tablet in USP or EP or IP, it will tell you what
Acceptable
are.
limit is given for essay, for example.
Yeah, and it tells you what test should be done for a tablet, what should test. So
Yes.
this is pharmacopoeial knowledge, okay?
Okay.
Now I am looking at my product composition and process which I have suggested,
Yes.
and with my knowledge of pharmaceutical processes, this is my justification, is my estimate, my anticipation, that this will be critical and this will not be critical. And I am building some theoretical pharmaceutical justification.
So when you're submitting data to regulator, this justification is acceptable to them, like with this.
Of course, ये तो starting है ना.
Right,
इसके बाद
okay.
I will do risk assessment.
Okay.
Then I will do experiments. Then I will show that all the risk is taken care
The
of.
design space and everything again has to
That
be done.
ये तो starting है.
Yeah, okay.
ये तो development का start है.
Okay.
तो लंका बहुत दूर है.
Yeah, okay.
Very far.
Thank you.
Okay. So basically, this is how to define the product strategy basically. Okay, similarly, so basically what you have done up till now is we have defined for ibuprofen what is the quality target profile that we want to achieve and what are the CQAs of the core tablet, of the coated tablet, you know, it may be different. For example, in the core granules, flow will be very critical, which you will not look at it in the dry mix, but when I am in a granule stage and when I want to operate on a very high speed machine, the flow and the particle size will be very critical. So you can define those and study those, the impact of those. Okay. And then if you look at risk assessment, so risk assessment typically, you know, there are many tools for risk assessment. Actually, I skipped that presentation. But so what you are looking at is the uncertainty, the severity if that case happens, and then so in the PHA model, probability hazard analysis, we look at the probability of the uncertainty and the severity. In the FEMA model, we also look at the detectability. So there are different models which are used, and you can, these are the two commonly used ones and very easy to use. So I will just see. One second, I'm just trying to see. Yeah. So basically in a PHA model, what you do is you look at the probability. Okay. Now probability in PHA, typically you have three, and these probabilities or scores is something that you can set on your own, based on your own logic. But this is normally what is done. So one is you have full knowledge. Okay. So when you have full knowledge, the parameter set point range limit has already been properly evaluated in the product. If it is being done, then you already know that this has to be what it is. Then the chances of any uncertainty is less than 1 in 200 chance of failure. Whereas prior experiences with some other product, if you have experience or other things, then it becomes medium. And then published literature, if you have some probability level of that failure happening, then that becomes level 3. And then 4 is insufficient knowledge. So when the effect of that parameter is not understood at all, and therefore the probability of the failure is very high. Against that you have severity levels, 1, 2, 3, 4. So severity, the parameter has no impact on the patient. So it's 1 level. 2 is it has some impact but reversible. 3 is there is an impact on the patient but not life-threatening. 4 is when there is an impact which is irreversible and potentially life-threatening. And when you put these in a matrix, then this is what it comes out. So you have probability 1, 2, 3, 4 and severity 1, 2, 3, 4. And so when you have low probability and low severity, the risk is very low. When you have medium probability but high, if the severity goes high, then it becomes high. Medium probability with low severity is medium risk. And when you have very high probability, severity high, even though the probability is low, it is high risk. Because the cause, severity means it can cause loss of life or some irreversible damage. So when the severity is high, it is all high. And so this is the matrix which is typically used. And it's very logical if you really consider that severity is given more importance. And then when both are high, of course it's a very high thing. When you cannot control the uncertainty or the probability of it happening and the severity. Now when you apply this, for example, over here, now this has been applied to the drug material attributes on product CQAs. Now the CQAs of the products which have been are the physical attributes, assay, content uniformity, dissolution and degradation. Okay. This is the CQA of the product. the finished product. Okay? And here you have the material attributes of the drug substance, ibuprofen. So for example, if you look at particle size, okay? Now the particle size physical attribute has a low risk. Uncertainty is true because you know the material or you know what is the impact of a similar material. Severity is one because it is something to be studied and to be controlled. But when you look at assay, the severity is very high. If the particle size is not proper, you may not get a proper assay, mixing may not be uniform. Similarly for content uniformity. And dissolution may particle size is of the high risk because in case of dissolution, it is totally dependent on the particle size for ibuprofen, which is a class 2 drug, it's a low solubility drug. Similarly, you have the impact of the hygroscopicity, the assay of the API, the related substance data of the API, you mean what is the actual impurity content of the API, the residual solvent, flow properties, solids and solubility. And going further, if you see, there is a reference given over there. So this is how you build the risk assessment. For example, if you say particle size, ESD is not expected to have any impact on the physical attribute, so risk is low. Assay, it will affect the flow, subsequently assay and uniformity and hence percentage in ibuprofen, and therefore it's a high risk, risk is medium. So like this, you define for everyone, you define a theoretical consideration why you are giving it low risk or high risk. And how do you arrive at this? You arrive at this risk analysis as a team effort. You actually sit and discuss with the team, okay, we know this is what happens in ibuprofen. We have also done olmesartan 2 years ago. It's a similar molecule with same sort of pKa. It's going to have the same thing. And that's why this is arrived after a discussion with the team. It's not arrived individually. And that's how you put it. So if you look at this, again, this is the impact of drug substance particle size on the CQAs. Hygroscopy, what impact it has. So it's non-hygroscopic photodegradation are not reported, API drug master file, etc. Hence risk is low. This information you have. So that's why I said before you make a strategy, before you do risk assessment, you need to collect all the information. If you already know that, it's not going to cause a problem. What's the point in studying it? Why waste resources? Understand where it's leading. Similarly, assay has no impact on physical attributes. But assay will have a direct impact on drug product, assay uniformity and resolution. Hence the risk is very high. If the assay is not within range of the API, you are not going to get a proper assay in the finished product. So similarly, like this, you build a justification. So basically, this is what you discuss and you build it. Okay, then this is again risk assessment of the impact of the formulation variables. Okay, so you have the disintegrant level, binder level, surfactant level, lubricant level. What impact it will have on? Now, this is something which all of us skilled in the art know, that if the binder level is not proper, you have a risk of physical attributes not being okay. You get sticking, picking, capping, whatever. So you will have this. It has an impact on disintegration and dissolution. So, I mean, for people like us who are skilled in the art of pharmaceutical sciences, you can draw this very easily. You can build a justification. Sometimes you know something and you put, for example, I think something about the melting point of the surfactant level is not expected to have any impact. But it will have direct impact on the release. So for drug release, the surfactant is very important for ibuprofen because it's a low solubility. So therefore, you come to this. So this is the PHA tool which has been used for studying the risk assessment of ibuprofen active substance and the composition, percentage of lubricant, the composition parameters on the product. And then for the process, here as an example, what we have is the FEMA. Now the difference in the FEMA is that you have the same uncertainty. Of course, in FEMA typically you have higher scores. So this is what we have formulated because you are multiplying and you are going to a certain number. So if you have very low scores, then it's very difficult to differentiate. So typically you have uncertainty, which is again full knowledge, prior knowledge, published literature, and insufficient knowledge. The severity is negligible, low, high, medium. Severity also same. But here again you have a detectability. So if the failure can be detected during unit operations, then it's a very high detectability. If it can be detected after unit operation but before end product testing, high. So it's good. Can be detected by end product testing, low detectability. But if there is no ability to detect, then this is a very severe case. If there is some failure which can happen and if you cannot detect it, then you are really in a very, very bad situation, and therefore that is something which you have to really think very deep about. So if you look at it and then what you do is you multiply the uncertainty factor, the probability, uncertainty, probability, severity and detectability, you come to an RPM number, which is called the risk probability number. And typically what happens is that if severity is more than 5, okay, and RPM is more than 5, then it's very high. If severity is more than 5 but RPM is less than 50, then it's medium. So it's all based defined on the severity, because the severity of the impact is very important. That is given key over the RPM. And then if the severity is less than 5, with RPM more than 50 is medium, with RPM less, the total product less than 50 is low. And therefore you measure the severity that way. So here just to give an example. Now if you look at selection of the API distribution. Now the API, selection of the API, source of API which you select, typically you study 2-3 sources. So it will have an impact on the assay and content uniformity. Causes of failure could be non-uniform distribution of the API. And then you will have, so basically what are the current controls which you have in place? So I am using micronized grade, particle size will be monitored, particle size specification is already decided, wet granulation process is selected. So based on this I am trying to control this probability. So severity of it failing is high, probability of happening is also high, but detectability is very low. So overall my score is 49 and therefore it's a medium risk because it is falling here in this. It's falling in this range. So it's a medium risk. Similarly, when I am looking at dry mixing of the blend, again improper distribution can happen. This is the failure which can happen. What is the tentative range of mixing is 5 to 15. Now currently I can detect it very high, but the severity is high and the probability is very high. So it is 70. Now this I have to optimize. Because I don't know. I am saying tentative range 5 to 15 minutes, but in 5 to 15 minutes will I get a uniform distribution of the API? I don't have that information at the start of the product. So I will study that. And what happens at the next step, again you say I have studied this range and I found that within 15 to 25 minutes, I am getting content uniformity well within the defined parameter. So my severity and my occurrence will come down. And therefore my risk number will go below 50 with the severity less than 5, so it will become low risk. That's how you do it step by step. So at the start of the product, but it's very important to think and put these risks on paper. So you know that I have to study this. Dry mixing time is critical. I have to study this and I have to optimize this and have to understand what is good for my product, which range is good for my product. Similarly, granulation. Now granulation can have an impact on all of these parameters. So kneading time speed to be optimized during development. Similarly for drying time again LOD very critical for flow and hardness and everything. So fluorescence, this will be optimized. Now look at the sizing milling. Improper sieve selection, 20 mesh and retention mill speed. It will be evaluated and finalized, but it's expected to have very low risk because this is known. For this sort of a tablet, this is known in the art. 500, 700 milligram tablet, if I use 20 mesh granules and if I control the particle size, I will most probably get a very good flow. This is known. So therefore you already know it and you are applying that knowledge. So you will say it is low. Blending and lubrication, again it's medium risk. Compression is always very high risk because you need to understand whether you can control at high speed the weights and everything. Hardness again is it has an impact on resolution profile. So again you need to be optimized. Coating again. So like that you go and you do this full risk assessment and then you start based on this you plan your experiments. First you will do experiments to set your composition parameter and then you will go to your process parameters. And after you have done your exercise, you will go back and do review. Again the risk, and like that you keep it's a multiple stage, and then when you do scale up also, again you do a risk assessment before scale up, after scale up, and then finally at the end of the day we want to come to all low risk, not zero risk, low risk. Okay, we have time.
Maybe
Or
5
your-
10 minutes more we can do.
Okay. I'll just show you one control strategies, just to so this is a control strategy. This is actually a file which has been submitted. So, this is a control strategy. This is the final strategy which we have arrived at after doing all the work. So, what we have is, we are talking of the raw material attribute. This is a tablet which is multiple strength dosage proportional. So, for the drug particle size distribution, this is the one which is decided, which was used in lab scale. This is the one which is used in the exhibit batch at the pilot scale. Pilot scale meaning 100,000 tablet batch, which we make registration batches for submitting, doing the study, bioequivalent study. So what has been used is 15 to 25. Proposed range is the same. And this, how am I going to control this? Through the drug substance specification. So the particle size of the API is very critical from the bioequivalent point of view. I've used something in development. I've used very narrow one in exhibit batch, and that's what I'm going to control going forward. So this is your control, and this is the type of control, how you're going to do it. So my drug control substance is going, specification is going to control the particle size, which is critical. For lactose, again particle size is critical because this is a direct operation product. So there is an in-house specification to control this. This is not part of the pharmaceutical specification. Especially there is an in-house specification to control the lactose particle size. You are using a certain period. HPC again, particle size distribution of the product, okay, attribute it controls. So what is the retention on the mesh during development, during scale up, and this is again in-house specification for the in-process. So after granulation, this is what, HPC, this is talking of the particle size distribution of the finished product and how you are going to control that. Then MCC, again particle size, this is particle size distribution of the HPC, this is a DC product. And again MCC, we have the particle size spec. Again particle size spec for all the excipients are very critical because this is a DC product. And the content of the API is very, very low. So we need to get content uniformity also. And then we have the magnesium stearate, and the surface area we think is critical and it is controlled. Then the dry mixing time, what is the dry mixing time, binder addition time, oh it's very easy. Kneading time, impeller speed, chopper speed, what was used in lab scale, exhibit batch, and what will be used going forward. And it is controlled in the operating range, in the MFC, manufacturing formulation control. The MFC formula card or the BMR, batch manufacturing record, is controlled through that. Similarly, drying process parameters, milling parameters, pre-lubricated blend, what are the in-process parameters, how they will be controlled. Then you have the in-process controls again of the during compression. What are we going to control, how we are going to control for the different strengths. And then coating, again in-process specification. So basically, and then this is the release specifications for all the products. So after all that work which we do in development, scale up and all, we come up with this sort of a control strategy. Now this is a fixed manufacturing process. You can also have a flexible manufacturing process. You can have a flexible range if you do the design space experimentation. Okay. So that is what I wanted to show you. I can show you the design space. Everybody is very tired. No man. Design space का really अच्छा होगा. Okay, let me show you this DOE example, how DOE has been used. Okay, so here is optimization and level of disintegration. So based on preliminary formulation development studies and initial risk assessment, Formulation variables identified which can impact this drug product CQA are intragranular and extragranular. So intragranular there is cross-terminal, extragranular is cross-terminal. This is again a almost class 4 drug, if I'm not mistaken. Very, very difficult to get bioavailability sort of drug. And the disintegrant is playing a very critical role. So what we have done is, see, we have the disintegrants, we have got the level 1, -1, 0, -1 plus 1, -1 plus 1, 0 and -2. So this is added at 2% and we are having levels -1, -2. So percentage weight by weight, so this is 9.56, we have studied 7.5, 9.5, 11.5. Here this is 3.82, 1.8 and 5.8. And this is the milligram per capsule. Now, so this is the factors which we are studying, what impact they have. Now the response which is critical and which we know because we have done the development, we have done a pilot study, we know what is critical in the product, so we apply that. So what is critical is this release in pH 4.5 acetate buffer. This is very critical because this is related to it's a biorelevant method. It's related to the pharmacokinetic data. So here we know that we should have not less than 40% at 120 minutes. This is the key. It doesn't go to 100%, but if I have less than 40%, I am not going to get a product which is bioequivalent to the innovative product. I know that. And of course, disintegration time should be not more than 30. Because it's a disintegrant, we are looking at that. So these are the trials as per the center point method. And these are the levels. And these are the trials for the other disintegrant. And we have done the physical evaluation of all these batches. Okay. So this is all the data is collected. Now we have done the ANOVA analysis. And what happens is that model F value of 3.92 implies that there is a 7.99 if you see the model F value. This is the F value. Here, model value, here. F value. So it implies that there is a 7.99% chance that model F value this large could occur due to noise. And probability less than 0.05 indicates model terms are significant. But in this case, there are no significant model terms. So value greater than this indicates that the terms are not significant. So we have not got a significant difference basically. And similarly is for the fit value. So, but it means that the ratio, it can be used to navigate the design space because the signal to noise ratio is greater than 4 is desirable. But what we have got is actually 3.85. When we look at the graphical representation, this is cross terminals and this is cross corridor. So this is where you have got the midpoint. This is quite right in the middle. This is when we are looking at dissolution of at 4.5%. And this is the contour plot. Similarly, when we look at the numerical optimization, what I am getting is from this numerical optimization, what it says is, from the desirability plot, the solution provided was cross-terminals concentration of 9.63 and cross-corridor of 2.8. This is what we are coming here. And it's very close to the center point. This is the center point of 9.56 and 3.82. So whatever we have selected is actually very close. We have studied the range plus minus 2% of both. But whatever is theoretically selected as 100% is actually very close to what as per the desirability plot satisfies or gives a product complying to the response factors. And when this is the overlay plot. So this is telling me that other than this, any concentration of either cross-terminals from here to this point or cross-corridor, sorry, from this point to this or this to this, it will give me a product which will comply. Okay, but the best is this, where you have 9.63 and 2.89. This is the one which is within the desirable range statistically. And therefore, what happens is that this is the design space which we have arrived at. We have studied this. And this is the design space which we have arrived at. Now, this means that when I scale up tomorrow, I can play within these ranges. I can change this without having to generate data because what this statistically tells me is as long as my sodium is between this range and cross-corridor within this range, I will still get a product which is meeting my critical attribute which I have identified. If I have identified a wrong attribute, then God save me. Okay, that is always there. So you need to understand, you need to apply and understand what is critical. Requires a lot of introspection and data analysis and brainstorms. So, I mean, it's not as simple as it looks, but this is how it is applied. And then, of course, you do the updated risk assessment and you say it's well. So this was one of the critical composition parameters. So I think we will close for the day because examples will never fall there, but I'm sure you must be very tired. Listening to people all day long. So if there are any questions, I'll be ready to take.
Please, Madam take the chair. He would like to summarize.
Yeah, sure.
Thank you so much, madam, for your very enlightening session on quality by design and your vast experience in the industry and your industrial expertise. Well explained the various concepts, explained about the historical perspectives, thereafter the different focus on product quality, continuous shift in quality paradigm. They also explain the importance of ICH Qpd guidelines in pharmaceutical product development with respect to quality target product profile, critical quality attributes, critical material attributes, etc., including the design of experiment. They also explain the ICH Q9 quality risk management aspect associated with the risk assessment, control, review, and the knowledge of the risk assessment. You also explain about the ICH Q10 guidelines of pharmaceutical quality system, which focuses on the continuous improvement in the product. ICH Q12 regarding the technical aspects, consideration for the pharmaceutical product life cycle management. We have talked about the concept of quality by design, what is quality by design, and critical aspects of the practical quality by design. Common causes and special causes of variation. Explain what is the rework and what is currently or where currently the industry today. Various Qpd life cycle approaches. Can explain the various elements of quality by design like Qtpp, critical quality attributes, critical material attributes, and explain about the product design understanding and process design and understanding. They have explained how the knowledge management is important to any organization or for the growth of any organization. With your very good or very what ma'am also said, the edited book of process development, product development, they have explained about the various concepts of quality by design by giving a lot of examples of quality target, product profile, critical attributes, and at what different level we need to consider these different aspects in the development of the product, including the control strategy. Even that also we have given a lot of examples. So it was a nice session, and you have explained about the different aspects that we need to be as a part of quality by design aspect, and which has been followed in the industry with different number of considerations. So thank you so much, madam. So we take the questions from the participants. So if you have questions, you can ask. Madam, I have one question. So you are there from a long time in industry, and you are there entrepreneur, and you have been a second entrepreneur right now. There are a lot of regulatory agencies in pharmaceutical industry, and if someone wants to start his own startup, first he look towards these are n number of regulatory bodies and how we can survive. So I just want to know your experience or how you faced about all of these and a successful entrepreneur and with this huge experience.
So, I think if we understand that the regulatory requirements are there for a certain purpose, and like I said, the guidelines are actually very helpful because they explain us exactly how a regulator thinks, and it's of course it helps a lot in understanding and then applying it so that it can help you justify whatever you are doing.
Although having said that, I think it's challenging sometimes because regulations keep on changing and the bar keeps on going up. Business realities are also there which we have to tackle. And also when we have audits, my observation is that each auditor is a different animal. Somebody gets stuck on micro, somebody gets stuck on stability, somebody is actually a analytical chemist, तो वो जाके HPLC के पास ही बैठ जाएगा audit trail करने को. And whatever you do, you know, finally we are human beings and we make mistakes. So to get that quality mindset and thinking, for people to, you know, you have sometimes like 200, 300 SOPs in a company and people are expected to remember and follow that. Train people to think everything, कि ये हमारे SOP के हिसाब से ऐसे नहीं है, ऐसे करना चाहिए. And somebody to keep on guiding and cajoling them कि ऐसे मत करो, चलो अब ये deviation हो गया, deviation बढ़ाओ. It is very difficult because the normal human tendency is push it under the carpet. तो वापस deviation कौन भरेगा यार मेरा कुछ ही बच गया. So this is a problem. The quality mindset is very difficult, and you can only build that if you have very good leadership. You get a lot of. If you have an environment where there is no fear of failure or fear of making a mistake. Because leaders we understand that human beings are there and they will make a mistake. And why they are making mistake? Either because they are overloaded with work, or they don't understand what they want to do, or they just cannot cope with the work pressure. Then as a leadership you have to try to take care of those things, you know. I think my personal view is even when I was CEO at Rubicon and we had a running plant which was approved and I used to always go with my team and tell them and ask them, what is the problem? Why are you not able to follow the SOP? Tell me the real problem. Don't tell me mara sahavas hai. Aisa nahi, tuna padha nahi, ki you thought it was not important, or you didn't realize that what you are doing is you are actually going against whatever is this norm. What is it? Because unless you tell me exactly, we cannot help you. If you don't understand, we can explain. If you need training, we can train. If you need to go out for training, we can send you. But if we don't know what is the problem, we cannot solve it. And in order for people to be able to frankly tell you, ki, Baba, I have been told to do something and I don't understand, it means it reflects poorly on their boss and it's very difficult. So to have that open environment to say, everybody makes mistakes, I also make mistakes, no problem, we'll correct it. But you should realize that a mistake is made and don't make the same mistake again. Now to build that sort of an environment in our sort of a hierarchical organization is very difficult. So as long as you have a small team, it's very easy. It's a flat team. But the moment you become big, it becomes very difficult to have that environment. And I think that is the main challenge. Because regulations are very, very logical. Finally, they are there to ensure the safety of the patient. So I don't see a problem with that. But every guideline or every requirement has to be thought through and applied. You cannot apply it blindly. Is it really suitable or applicable to your product and your patients and all? So, वो एक जो basic macro level thinking करके you have to bring it down and then apply it. That requires a lot of effort. For example, when we set up Rubicon, we had a consultant and all, but we had our own team and a very senior person. And you were getting actually cut paste करके SOPs सब मिलता है, you know. Typical, there are standard SOPs for QC lab and this and that and all. But what we did is we spent 1 year on it because we sat with our team and we said flow sheet banao. If you have to control the quality of your raw material or if raw material has to come into your warehouse, what is the procedure you will follow? What is the flow sheet? What is the risk which can go wrong? You draw a flow sheet and build your own SOP. Now this took us a long time, but we built SOPs exactly to what you were following. So finally, what is for an audit? What is important is you do as you say and you say as you do. You do exactly what your SOP says and you say exactly in your SOP what you are doing. And if you change it, modify your SOP. That is totally in your control. So this going from the basic and then building the system and building the field, it's very, very time consuming. It requires a lot of effort and it's very difficult to make 50-100 people think the same thing. Everybody's mind is individual here. So ये बहुत बड़ा challenge है industry में. building that quality culture or quality mindset and everybody committed to it, it really requires a very high level of
So, madam, very exhaustive but very interesting presentation. And we pharmaceutics teachers understood because in our research we are applying Qpd. We have chemistry background people also. Probably a lot of pharmaceutics examples they might have got little difficult in correlating, but the concept they have understood, I am very sure.
No, I can give you a chemistry synthesis example. Which is that I don't have anything ready because it's very simple, yeah. I want to synthesize paracetamol. My starting material is A, B, C. And this is my synthetic process. अभी वो material में क्या चाहिए, process में क्या चाहिए, what you will control in the process, and what you will do in your recrystallization. See, it's the same concept which you have to apply. The concept doesn't change.
And also you have shown two products at the end. One was with the design space that you determined using design of experiment, and the previous one was you said fixed composition, so you have not used this design of experiment. So is it that design of experiment is not mandatory for quality by design?
You can do your full development, risk assessment, control strategy with one factor at a time. हम जो typical करते हैं ना one factor at a time experiment. Design of experiments is not mandatory, and design of experiments is not Qpd. It is only a tool.
So even regulators will not insist?
No, they don't insist. No, they don't insist. You go by your data and your science. If your science is strong, everybody has to agree with you. Your data should show that confidence in the process and the method. You don't have to do DA.
Thank you, madam. Any other questions? So, with this, now I request respected Dr. Supriya Shilay, madam, Principal Viscop, to come on the stage and felicitate Maharukh madam by giving the memento and certificate as token of appreciation.
Lot of information here.
Thank you so much, madam.
Ma'am's lectures are available on our YouTube channel, GS College of Pharmacy YouTube channel. She was a resource person for our program for transition of a student from undergraduate stage to postgraduate research scholar. In that series, her lecture is there. Please do watch that lecture. It is really very informative. Whenever I get time, I go back to that lecture and revise my certain. It was about pharma industry. Okay. You can also watch it. I forgot what I taught.
Yeah, I always try to start with the industry figures.
It was
our
online,
students,
that program.
nobody looks at that. Ki how big is the industry? Where is it going? Which are the therapeutic areas? Wellness industry, nutrition industry. They all need our technologies. So it's interesting.